Hello YouTube, my name is Neil and we're studying for the MCATs today. We're in the Biochemistry review, Chapter 5, Lipid Structure and Function. What do beer, human eyes, and sperm whales have in common? For all of these, lipids play a key role in some of the most interesting characteristics. The taste and smell of hops comes from lipids called terpenes. The ability of the human eye to respond to light and therefore see relies heavily on retinol, a lipid derived from vitamin A. Sperm whales use an enormous reservoir of spermaceti to dive to a depth of up to 3 kilometers to hunt giant squid. Spermaceti is a lipid with a density that changes dramatically with temperature, effectively allowing sperm whales to adjust their density with depth so that they can stay submerged without having to constantly fight buoyancy. Lipids as a class are characterized by insolubility in water and solubility in nonpolar organic solvents. Aside from this shared feature, lipids diverge dramatically in their structural organization and biological functions, serving vital structural, signaling, and energy storage roles. In this chapter, we will explore the structural and functional characteristics of each of the major categories of lipids tested on the MCAT. As structural building blocks, we will investigate phospholipids and sterols, which make up vesicles, liposomes, and membranes. When it comes to signaling, we will note that lipids serve multiple roles, from enzyme cofactors to light-absorbing pigments, and from intracellular messengers to hormones. Finally, we will see that lipids are the workhorse of energy storage, giving the most bang for the metabolic buck by weight. 5.1 Structural Lipids. Structural. Lipids are the major component of the phospholipid bilayer, one of the most important structural parts of the cell. The unique ability of phospholipids to form a bilayer allows a cell to function as they do, separating the cell interior from the surrounding environment. We will first take a close look at the structure and role of phospholipids, Glycerophospholipids, and Sphingolipids. Finally, we will review the gross structural characteristics of the unique class called waxes. Each of the membrane components in an amphipathic molecule, meaning that it has both hydrophilic and hydrophobic regions. For these membrane lipids, the polar head is the hydrophilic region, whereas the fatty acid tails are the hydrophobic region. When placed in aqueous solution, these molecules spontaneously form structures that allow the hydrophobic regions to group internally while the hydrophilic regions interact with water. This leads to the formation of various structures, including liposomes, micelles, and the phospholipid bilayer shown in figure 5.1. Remember that amphipathic means hydrophilic and hydrophobic portions. Regions. And let's draw a little phospholipid. Actually, we do this. We'll do, we got the heads that are hydrophilic. They like water. We got the tails that are hydrophobic. All right. Phospholipids. Phospholipids contain the following elements: a phosphate and alcohol that comprise the polar head group joined to a hydrophobic fatty acid tail by phosphodiester linkages. Phospholipids include a phosphate, alcohol, is the head, and then the fatty acid tail. One or more fatty acids are attached to a backbone to form the hydrophobic tail region. Phospholipids can be further classified according to the backbone on which the molecule is built. For example, glycerol, a 3-carbon alcohol, forms phosphoglycerides or glycerophospholipids. And sphingolipids have sphingosine backbone. One important thing to note, however, is that not all sphingolipids are phospholipids, as described later in this chapter. The backbone is an alcohol. Sphingolipids or glycerophospholipids. So they're still like, so this alcohol part is the head, is this alcohol. A phosphate, glycerol, and then a fatty acid tail. They didn't tell us what the fatty acid tail looks like. We'll talk about it. One thing that these lipids do all share in common is a tail composed of a long chain fatty acid. These hydrocarbon chains vary by their degree of saturation and length. These two properties determine how the overall molecule will behave. Fully saturated fatty acid tails will have only single bonds. The carbon atom is considered saturated when it is bonded to 4 other atoms with no pi bonds. Saturated fatty acids, such as those in butter, have greater van der Waals forces and a more stable overall structure. Therefore, they form solids at room temperature. An unsaturated fatty acid includes 1 or more double bonds. Double bonds introduce kinks into the fatty acid chain, which makes it difficult for them to stack and solidify. Therefore, unsaturated fats like olive oil tend to be liquids at room temperature. The same rules apply in the phospholipid bilayer. Phospholipids with unsaturated fatty acid tails make up more fluid regions of the phospholipid bilayer. Phospholipids, glycerophospholipids, and sphingolipids can have any of a variety of fatty acid tails and also different head groups, which determine their properties at the surface of the cell membrane. The next 2 sections, glycerophospholipids and sphingolipids, focus on the various polar head groups that different phospholipids may have. So the head and the tail is just hydrocarbons. Glycerol makes glycerophospholipid, and sphingosine makes sphingolipids. I guess the phosphate makes the phospholipid. And saturated and unsaturated. I guess if we have a head And we have a long carbon chain, and they're all just hydrogens. It's pretty saturated. On the other hand, unsaturated would be missing hydrogens, causing double bonds between carbons. I guess they wouldn't be right next to each other. They'd be conjugated. And then it could kind of continue that structure. And maybe there's another kink down here. There you go. One, two, three, four. 1, 2, 3, 4, 5, 6, 7. I made 7 carbons over here. 1, 2, 3, 4, 5, 6, 7. $16 hydrogens on this one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 hydrogens on that one. All right. Glycerophospholipids. As mentioned in the last section, glycerophospholipids are all phospholipids, yet not all phospholipids are glycerophospholipids. Glycerophospholipids, or phosphoglycerides, are specifically those phospholipids that contain a glycerol backbone bonded by ester linkages to 2 fatty acids and by a phosphodiester linkage to a highly polar head group. As shown in figure 5.2. Because the head group determines the membrane surface properties, glycerophospholipids are named according to their head group. For example, phosphatidylcholine is the name of a glycerophospholipid with a choline head group. And phosphatidylethanol ethanolamine. Ethanol. Phosphatidylethanolamin. Ethanolamine. Phosphatidylethanolamin. There we go. Ethanolamine. Phosphatidylethanolamin. Oh, that's a fun word to say, huh? Phosphatidylethanolamin is one with an ethanolamine head group. Let's write these down. Phosphatidylcholine. Ethanolamine. Phenylalanine. The head group can be positively charged, negatively charged, or neutral. The membrane surface proteins of these molecules make them very important to cell recognition, signaling, and binding. Within each subtype, the fatty acid chains can vary in length and saturation, resulting in an astounding variety of functions that are the focus of active scientific research. So ester linkage to fatty acids. And phosphodiester linkage to head group. Oops. Structure of glycerophospholipid. The polar head group is a phosphate and something. They said these are the ester bonds, and this is the phosphodiester bond. We're going to draw this as well. Great. Whoops. Polar, non-polar. Great. Sphingolipids. Blood typing makes it possible to give life-saving blood transfusions without risking potentially fatal acute hemolytic reactions. The ABO blood type system is based on cell surface antigens on red blood cells. These cell surface antigens are some of the most well-known sphingolipids. Like glycerophospholipids, sphingolipids are also sites of biological recognition at the cell surface and can be bonded to various head groups and fatty acids. Whoops. Sphingolipids. Cell signaling. Like blood type antigens. Antigens. cell surface antigens. Signaling. Sphingolipids have a sphingosine or sphingosine-like backbone, as opposed to the glycerol backbone of Glycerophospholipids. So I should write this down. This is the backbone here, right? No, this is the glycerol. This isn't the backbone. These two are the... I guess this whole thing is the backbone, right? Oh, this is nonpolar. This is the acid, fatty acid. This is the polar head. This is the backbone, right? So a sphingosine-like backbone, opposed to glycerol. These molecules also have long-chain nonpolar fatty acid tails and polar head groups. Many sphingolipids are also sphingolipids because they contain a phosphodiester linkage. However, other sphingolipids contain glycosidic linkages to sugars. Any lipid linked to a sugar can be termed a glycolipid. Sphingolipids are divided into 4 major subclasses, differing by their head group. Let's just throw glycolipid in there. Have glycosidic link to sugar. Sphingolipids are divided into 4 major subclasses. The simplest sphingolipid is ceramide, which has a single hydrogen atom at its head group. A single hydrogen atom, ceramide. Its head group is just H. Sphingomyelins are the major class of sphingolipids that are also phospholipids. Sphingospholipids. Sphingomyelins. These molecules have either phosphatidylcholine or phosphatidylethylamine as a head group, and thus contain a phosphodiester bond. Sphingomyelin head groups have no net charge. As the name implies, sphingomyelins are major components in the plasma membranes of cells producing myelin, oligodendrocytes, and Schwann cells, the insulating sheath for axons, right? Sphingomyelins. The head is either these two, phosphatidylcholine or phosphatidylethylamine. No net charge. Neutral charge. Sphingolipids with head groups composed of sugars bonded by glycosidic linkages are called glycolipids, as mentioned above, or more specifically, Glycosphingolipids. These molecules are not Phospholipids because they contain no phosphodiester linkage. Glycosphingolipids are mainly found on the outer surface of the plasma membrane and can be further classified as cerebrosides or globosides. Cerebrosides have a single sugar, whereas globosides have two or more. These molecules are also referred to as neutral glycolipids because they have no net charge at physiological pH. So should we write just glycolipids or properly Glycosphingolipids? Glycosidic linkage to sugar. They're also neutral. Cerebrosides have single sugars. Globosides have multiple sugars. The final group is composed of the most complex sphingolipid. Gangliosides are glycolipids that have polar head groups composed of oligosaccharides with one or more N-acetylneuramic acid, NANA, also called sialic acid. Molecules at the terminus and a negative charge. These molecules are also considered glycolipids because they have a glycosidic linkage and no phosphate group. Gangliosides play a major role in cell interaction, recognition, and signal transduction. Great. Gangliosides. They have a head of oligosaccharides plus one or more N-acetylneuramic acid. NANA. And we have this very faint figure of different sphingolipids. Sphingosine, ceramide, sphingomyelin, cerebroside, ganglioside. We have the hydrocarbon chain kind of in common. And then we have, I guess, ethanolamide in the case of sphingosine. In the case of ceramide, you have a fatty acid residue here. Sphingosine has no fatty acid residue. Ceramide has a fatty acid residue. Sphingomyelin has a fatty acid residue and a phosphocholine. Sphingomyelin, another sphingomyelin with another instead of phosphocholine, it's phosphoethanolamine. A cerebroside has a sugar. A ganglioside has a large sugar, an oligosaccharide. And we have sialic acid down here with another sugar and acid on it. COO. All right. Should I draw one of these? You can see the pattern, so I'm going to draw the basic sphingosine backbone. Let's see how many carbons there are. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. And then a double bond, then a single bond, and then a single bond, and then a single bond, and then OHs, NH3, oops, NH3 plus. That looks pretty good. There's sphingosine. And then we could add a fatty acid residue to this. And we'd have to knock off some of these hydrogens. And then we could also knock off this hydrogen over here. You know what, I'm going to do that in a different color. And then add some phosphates to this, or a sugar. Whoops. P O O. That would be your phosphocholine. You could also have phosphoethanolamine or a sugar. There's a single sugar residue, multiple sugar residue. But that would go in that green spot as well. So we're just going to leave that out. Next, waxes. Waxes are esters of long-chain fatty acids with long-chain alcohols. As one might expect, they form pliable solids at room temperature, what we generally think of as wax. Biologically, they function as protection for both plants and animals. In plants, waxes are secreted as a surface coating to prevent excessive evaporation and to protect against parasites. In animals, waxes are secreted to prevent dehydration as a water repellent, to keep skin and feathers dry, and as a lubricant. For example, carnauba wax is made from the leaves of the Copernicia prunifera palm and is used to coat candies and wax cars. Bees secrete waxes to construct shelter, as shown in figure 5.8. Wax. Long chain fatty acid plus long chain alcohol. Great. Which components of membrane lipids contribute to their structural role in membranes? Which components contribute to function? Structural and function. I guess that the polar head and the nonpolar tail. I guess that is the basis for the structure, or is the backbone the structure? I mean, the backbone holds together the polar and nonpolar regions, so sure, we'll call that the structure. But the fact that there's a polar and a nonpolar side is what makes the structure, but the function, I guess, because it separates water. But the polar head is going to have your function like signaling, like the sugars and the oligosaccharides. I think those are going to be used in signaling. The nonpolar side's really just the structure. Let's see what they say. What is the difference between a sphingolipid that is also a phospholipid and one that is not? A sphingolipid that is a phospholipid just has a phosphodiester bond. A sphingolipid that isn't a phospholipid is one of the ones that don't have phosphodiester bonds, or the ones linked to sugars have glycosidic linkages. So cerebrosides and globosides. Glycosphingolipids and gangliosides do not have are not phospholipids. Name the three main types of sphingolipids and their characteristics. Ceramide has just hydrogen as the head. Sphingomyelins have phosphatidylcholine and phosphatidylethylamine heads, and glycosphingolipids have sugars for heads. The ceramide and the sphingomyelin are both phospholipids, while the glycosphingolipid is a glycolipid. What would happen if an amphipathic molecule were placed in a nonpolar solvent rather than an aqueous solution? If it was nonpolar, then I imagine the hydrophobic part would interact, and the hydrophilic part would make the inside of the lipid bilayer. So you'd have like a reverse lipid bilayer. Let's see if that's what they imagine would happen. 5.1. Membrane lipids are amphipathic. They have hydrophilic heads and hydrophobic tails, allowing for the formation of bilayers in aqueous solution. The fatty acid tails form the bulk of the phospholipid bilayer and play a predominantly structural role. On the other hand, the functional differences between membrane lipids are determined by the polar head group due to the constant exposure to the exterior environment of the phospholipid bilayer. Remember, this can be either the inside or outside of the cell. The degree of unsaturation of fatty acid tails can also play a small role in function. Oh, there you go. The amount of saturation of the nonpolar part can have small function in like how permeable the membranes are and shit, right? The difference is number 2, the difference is the bond between the sphingosine backbone and the head group. When this is a phosphodiester bond, it's a phospholipid. Non-phospholipid sphingolipids include glycolipids, which contain glycosidic linkages to sugar. The 3 types of sphingolipids are sphingomyelin, which has a phospholipid, and has either a phosphatidylethanolamine or a phosphatidylcholine head. The glycosphingolipid is a glycolipid. They didn't put ceramide in there. They just went straight to sphingomyelin. I said ceramide, sphingomyelin, glycosphingolipid. They said sphingomyelin, glycosphingolipid, ganglioside. So the bottom two are glycolipids. It's just whether they have a single sugar or an oligosaccharide in their functional group. So what's the difference between a polysaccharide and an oligosaccharide? They are the same thing, right? Polysaccharide versus oligosaccharide. The main difference between oligosaccharides and polysaccharides is that oligosaccharides are made out of a few number of monosaccharides, whereas polysaccharides are made out of large numbers of monosaccharides. So what was it, like less than 20? Or was it like less than a dozen? Less than 12? Number 4, in a nonpolar solvent, we would see the opposite of what happens in a polar solvent like water. The hydrophilic polar part of the molecule would be sequestered inside, while the nonpolar hydrophobic part of the molecules would be found on the exterior and exposed to the solvent. Great. Signaling lipids. In addition to passive roles in structure, lipids also perform active roles in cellular signaling and as coenzymes. Lipids serve as coenzymes in the electron transport chain and in glycosylation reactions. Lipids also function as hormones that transmit signals over long distances and as intracellular messengers responding to extracellular signals. Certain special lipids with conjugated double bonds absorb light, which is extremely important for our ability to see. Others act as pigments in plants and animals. Here we focus on 3 important categories of signaling lipids: steroids, prostaglandins, and fat-soluble vitamins, as well as important precursors like terpenes. Terpenes and terpenoids. Terpenes and terpenoids. Before we delve into the details of downstream lipid signaling molecules, we must first turn our attention to terpenes. These odoriferous chemicals are the metabolic precursors to steroids and other lipid signaling molecules and have varied independent functions. Terpenes are a class of lipids built from isoprene, C5H8 moieties, and share a common structural pattern with carbons grouped in multiples of 5 as shown in figure 5.5. Isoprene is the most basic terpene, C5H8, and it looks like this. Terpenes are produced mainly by plants and also by some insects. They are generally strongly scented. In some cases, these pungent chemicals are part of the plant or insect's protective mechanism. The strong smell of turpentine, a derivative of resin, comes from the monoterpenes that are resin's major components. Terpenes actually get their name from their original discovery in turpentine. Terpenes are also the primary components of much more pleasant smelling essential oils extracted from plants. Terpenes are grouped according to the number of isoprene units present. A single terpene unit contains 2 isoprene units. Monoterpenes, which are abundant in both essential oils and turpentine, as described above, contain 2 isoprene units. A single terpene unit contains 2 isoprene units. So monoterpenes are 2 of these. So basically 2 isoprenes. Sesquiterpenes. Sesqui meaning 1.5 means 3 isoprene units. Sesquiterpene. So it's got to be a C15, H24, is that right? And diterpenes contain 4. And let's do that math really quick as well. 32. Vitamin A, which will be discussed later in this chapter, is a diterpene from which retinol, a visual pigment vital for sight, is derived. Triterpenes with 6 isoprene units can be converted to cholesterol and various steroids. Six isoprene units. So C30, 6 times 5, and 6 times 8 is what, 48? Cholesterol and steroids. Retinol, vitamin A, all right, carotenoids like beta carotene and lutein are tetraterpenes and have 8 isoprene units. Natural rubber has isoprene chains between 1,000 and 5,000 units long and is therefore considered a polyterpene. Tetraterpenes are going to have like C40. 8 isoprene units. 8 times 8 is 64. And rubber is also down there. So let's just write C1000. C5000, H8000. Great. That's rubber. Terpenoids, also sometimes referred to as isoprenoids, are derived of terpenes that have undergone oxygenation or rearrangement of the carbon skeleton. These compounds are further modified, as are terpenes, by the addition of an extensive variety of functional groups. Terpenoids share similar characteristics with terpenes in terms of both biological precursor function and aromatic properties, contributing to steroid biosynthesis as well as the scents of cinnamon, eucalyptus, camphor, turmeric, and numerous other compounds. Terpenoids are named in an analogous fashion, with diterpenoids deriving from 4 isoprene units and so on. Terpenes and terpenoids are precursor molecules that feed into various biosynthesis pathways that produce important products, including steroids, which have widespread effects on biological function, and vitamin A, which is vital to sight. Terpenoids are terpenes, oxygenated terpenes. Terpenoids are oxygenated or otherwise modified terpenes. Steroids. The term steroid probably brings to mind muscle-bound bodybuilders or home-run-hitting professional athletes. In science and on the MCAT, steroid refers not just to the infamously abused anabolic steroids, but also to a broader class of molecules defined by their structure. Structural steroids, shown in figure 5.6, are metabolic derivatives of terpenes and are very different from the lipids mentioned earlier in this chapter in both structure and function. Steroids are characterized by having 4 cycloalkane rings fused together, 3 cyclohexane, and 1 cyclopentane. Steroid functionality is determined by the oxidation status of these rings, as well as the functional groups they carry. It is important to note that the large number of carbons and hydrogens make steroids nonpolar, like the other lipids mentioned. Steroids are 3 cyclohexanes and 1 cyclopentane. 3 hexagons and plus 1 pentagon. Arranged like this, why not? Whoops. And then the pentagon. Great. Keep in mind the terminology difference between steroids and steroid hormones. Did I already read this? You should also just note that they're non-polar. Steroid refers to a group defined by a particular chemical structure, demonstrated above. Steroid hormones are steroids that act as hormones, meaning that they are secreted by endocrine glands into the bloodstream and then travel on protein carriers to distant sites where they can bind to specific high-affinity receptors and alter gene expression levels. Steroid hormones are potent biological signals that regulate gene expression and metabolism, affecting a wide variety of biological systems, even at low concentrations. Some important steroid hormones include testosterone, various estrogens, cortisol, and aldosterone, which are discussed in Chapter 5 of Biology. Plants, like animals, also use steroids as signaling molecules. Steroid hormone are effective at low concentration. Some examples: testosterone, estrogen, cortisol, aldosterone. Cholesterol, shown in figure 5.7, is a steroid of primary importance. Cholesterol is a major component of the phospholipid bilayer and is responsible for mediating membrane fluidity. Cholesterol, like a phospholipid, is an amphipathic molecule containing both hydrophilic and hydrophobic components. Interactions with both the hydrophobic tails and hydrophilic heads of phospholipids allows cholesterol to maintain relatively constant fluidity in cell membranes. At low temperatures, it keeps the cell membrane from solidifying. At high temperatures, it holds the membrane intact and prevents it from becoming too permeable. Cholesterol also serves as a precursor to many important molecules, including steroid hormones, bile acids, and vitamin D. So here is a picture of cholesterol. Let's draw it out. Cholesterol is made up of your steroid backbone with a carbon and an oxygen and a alkane tail here. So this is probably your this is going to be your non-polar side. I guess this oxygen here, this alcohol, is going to be your polar side. Cholesterol, we start with your basic cholesterol. Then you add an OH. We'll add a carbon here. We're going to ignore the hydrogen. We're going to add a carbon here. And on the top of this, we're going to go 1, 2, 3, 4, 5. Split. And then another carbon there. Cholesterol. Cholesterol is also in hormones. Bile and vitamin D. Prostaglandins. Prostaglandins acquired their name because they were first thought to be produced by the prostate gland, but have since been determined to be produced by almost all cells in the body. These 20 carbon molecules are unsaturated carboxylic acids derived from arachidonic acid and contains one 5-carbon ring. They act as a paracrine or autocrine signaling molecule. In many tissues, the biological function of prostaglandins is to regulate the synthesis of cyclic adenosine monophosphate, CAMP, cyclic AMP. which is a ubiquitous intracell messenger. In turn, cyclic AMP mediates the actions of many other hormones. Downstream effects of prostaglandins include powerful effects on smooth muscle function, influence over sleep-wake cycles, and the elevation of body temperature associated with fever and pain. Non-steroidal anti-inflammatory drugs, NSAIDs, like aspirin, inhibit the enzyme cyclooxygenase COX, which aids in the production of prostaglandins. Prostaglandins arrive from arachidonic acid, paracrine, autocrine signaling. And cyclic AMP. So we also write down that NSAIDs target cyclooxygenase which aids in prostaglandin production. Fat-soluble vitamins. A vitamin is an essential nutrient that cannot be adequately synthesized by the body and therefore must be consumed in the diet. Vitamins are commonly divided into water-soluble and lipid-soluble categories. Lipid-soluble vitamins can accumulate in stored fat, whereas excess water-soluble vitamins are excreted through the urine. The fat-soluble vitamins include A, D, E, and K. Each of these has important and varied functions. Fat-soluble vitamins are A, D, E, and K. Vitamin A, or carotene, is an unsaturated hydrocarbon that is important in vision, growth, and development, and immune function. The most significant metabolite of vitamin A is the aldehyde form retinal, which is a component of the light-sensing molecular system in the human eye. Retinol, the storage form of vitamin A, is also oxidized to retinoic acid, a hormone that regulates gene expression during epithelial development. Vitamin A is also called carotene. Retinal. Retinoic acid is part of development. So this is for the eyes. This is for development. Epithelial development. What is epithelial development? During development, epithelial by epithelial development, do they mean in the early stages of life or just period, like throughout life? Development, not like growth and replenishment, but development implies at birth, right? Like that process. Vitamin A. Vitamin D, or cholecalciferol, can be consumed or formed in a UV light-driven reaction in the skin. In the liver and kidneys, vitamin D is converted to calcitriol, the biologically active form of vitamin D. Calcitriol increases calcium and phosphate uptake in the intestine, which promotes bone production. A lack of vitamin D can result in rickets, a condition seen in children and characterized by underdeveloped, curved long bones as well as impeded growth. So, vitamin D is cholecalciferol. It can be consumed or formed in a UV light-driven reaction in the skin. So something is taking a cholesterol and with the help of UV light turning it into vitamin D. In the liver and kidneys, vitamin D is converted to calcitriol. So it's turned into calcitriol in the liver and kidneys, which is the active form. Vitamin E. Characterizes a group of closely related lipids called tocopherols. Tocopherol. And tocotrienol. Tocotrienol. These are characterized by a substituted aromatic ring with a long isoprenoid side chain and are characteristically hydrophobic. Tocopherols are biological antioxidants. The aromatic ring reacts with free radicals, destroying them. This in turn prevents oxidative damage, an important contributor to the development of cancer and aging. They don't tell us what tocotrienols are, but tocopherols are antioxidants. And they have aromatic rings. Vitamin K. Vitamin K is actually a group of compounds including phylloquinone and menaquinones. Vitamin K is vital to the post-translational modifications required to form prothrombin, an important clotting factor in the blood. The aromatic ring of vitamin K undergoes a cycle of oxidation and reduction during the formation of prothrombin. Vitamin K is also required to introduce calcium binding sites on several calcium-dependent proteins. Vitamin K, phylloquinone is K1, menaquinone is K2. There's a whole bunch of them. Prothrombin, clotting factor. Also aromatic ring. How many carbons are in diterpene? Diterpene sounds like 2, but it's actually 4 isoprene units, each of which have 5 carbons. So 20 carbons. What is the difference between a steroid and a steroid hormone? A steroid is just the structure. A hormone is a hormone based off of the structure. NSAIDs block prostaglandins production in order to reduce pain and inflammation. What do prostaglandins do to bring about these symptoms? Prostaglandins So prostaglandins activate the autocrine system The biological function of prostaglandins is to regulate the synthesis of cyclic AMP. In turn, AMP mediates the actions of other hormones. So, what do prostaglandins do to cause pain that the NSAIDs are shutting down? Cell signaling. Regulating cyclic AMP. Prostaglandins are part of cell signaling through the mechanism of cyclic AMP. NSAIDs block prostaglandin production in order to reduce pain. Oh, we just did that one. 4. What are the names and functions of the 4 fat-soluble vitamins? The fat-soluble vitamins are A, D, E, and K, and their function is to make retinol and retinoic acid. They make vitamin D makes calcitriol. Vitamin E makes tocopherol, which is an antioxidant. Vitamin K makes a bunch of things, including prothrombin, which is a clotting factor. What is the function of the vitamins? Well, the functions of the vitamins are to be a part of something else that's a signal or a hormone. That's kind of across the board. .2. A diterpene has 20 carbon molecules in its backbone. A steroid is defined by its structure. It includes 3 cyclohexane rings and a cyclopentane ring. A steroid hormone is a molecule within this class that also functions as a hormone, meaning that it travels in the bloodstream, is active at low concentrations, has high affinity receptors, and affects gene expression and metabolism. Prostaglandins regulate the synthesis of cyclic AMP, which is involved in many pathways, including those that drive pain and inflammation. 4. Vitamin A is involved in vision and epithelial development. Vitamin D, calcium and phosphate regulation, right. What does calcitriol do? Calcium uptake in the gut. Calcium uptake in gut. Vitamin E, antioxidants. Vitamin K, post-translational modification of prothrombin. Addition of calcium binding sites on many proteins. All right, so it's used in synthesis as well, protein synthesis. 5.3 is going to be energy storage. Energy storage. Triglycerols are a class of lipids specifically used for energy storage. From the body's point of view, lipids in general are a fantastic way to store energy. This is true for 2 major reasons. First, the carbon atoms of fatty acids are more reduced than those of sugars, which contain numerous alcohol groups. The result of this is that the oxidation of triglycerols yields twice the amount of energy per gram as carbohydrates, making this a far more energy-dense storage mechanism compared to polysaccharides like glycogen. Second, triglycerols are hydrophobic. They do not draw in water and do not require hydration for stability. This helps decrease their weight, especially in comparison to hydrophilic polysaccharides. One final perk for vertebrates surviving in colder temperatures like penguins, polar bears, and Arctic explorers is that the layer of lipids serves a dual purpose of energy storage and insulation. It helps to retain body heat so that less energy is required to maintain a constant internal temperature. Triglycerols. Triacylglycerols. It's not just triglycerols, it's triacylglycerols. Triacylglycerols, also called triglycerides, are composed of 3 fatty acids bonded by ester linkages to glycerol, as shown in figure 5.8. For most naturally occurring triglycerols, it is rare for all 3 fatty acids to be the same. So there'll be different fatty acids. So let's draw this out. We have CH2, whoops. Bonded to O. 1, 2, 3, 4, 5, 6, 7. Great. You got glycerol. And you got the fatty acids. Glycerol and fatty acids. All right. Overall, these compounds are nonpolar and hydrophobic. This contributes to their insolubility in water, as the polar hydroxyl groups of the glycerol component and the polar carboxylates of fatty acids are bonded together, decreasing their polarity. Triacylglycerol deposits can be observed in cells as oily droplets in the systole. These serve as depots of metabolic fuel that can be recruited when the cell needs additional energy to divide or survive when other fuel supplies are low. Special cells in animals known as adipocytes store large amounts of fat and are found primarily under the skin, around mammary glands, and in the abdominal cavity. In plants, triglycerol deposits are also found in seeds as oils. Triglycerols travel bidirectionally in the bloodstream between the liver and adipose tissue. The physical characteristics of triacylglycerols, are primarily determined by the saturation or unsaturation of the fatty acid chains that make them up, much like phospholipids. Free fatty acids and saponification. Let's first write down adipocytes, our fat cells. Free fatty acids and saponification. Free fatty acids are unesterified fatty acids with a free carboxylate group in the body. These circulate in the blood bonded non-covalently to serum albumin. Fatty acids also make up what we know as soap, which can be produced through a process called saponification. Saponification is the ester hydrolysis of triglycerols using a strong base. Traditionally, the base used is lye, the common name for sodium or potassium hydroxide. The result is the basic cleavage of the fatty acid, leaving the sodium salt of the fatty acid and glycerol, as shown in figure 5.9. The fatty acid salt is what we know as soap. So do you have to use triglycerols to make soap? Could you just use phospholipids? if you add a strong base to this, I wonder. Unesterified fatty acids. So what are unesterified fatty acids? So just this part without the glycerol. The free carboxylate group. So let's write down that fatty acids travel bonded to albumin. And saponification. We'd gone over that before in chemistry. Soaps can act as surfactants. A surfactant lowers the surface tension at the surface of a liquid, serving as a detergent or emulsifier. This is important to how soap works. If we try to combine an aqueous solution in oil, as with vinegar and olive oil in salad dressing, these solutions will remain in separate phases. If we were to add a soap, however, the two phases would appear to combine into a single phase, forming a colloid. This occurs because of the formation of micelles, tiny aggregates of soap with the hydrophobic tails turned inward and the hydrophilic heads turned outward, thereby shielding the hydrophobic lipid tails and allowing for overall solvation, as shown in figure 5.10. We saw these earlier when we discussed membrane lipids. Nonpolar compounds can dissolve in the hydrophobic interior of the water-soluble micelle, meaning that our cleaning agents can dissolve both water-soluble and water-insoluble messes and then wash them all away together. Micelles are also important in the body for the absorption of fat-soluble vitamins, A, D, E, and K. And complicated lipids such as lecithins, lectins, or lipolytic enzymes. So biosalts are like soap. Great. You got a picture of a micelle. How does the human body store spare energy? Why doesn't the human body store most energy as sugar? I guess there's more, you can store more energy in fatty acids because they're fully saturated. They could be more oxygenated. They're fully reduced, or they're fully oxygenated. Reduction is electrons. Oxygenation is bonds to hydrogen. So fatty acids are more oxygenated, so they could be further reduced, meaning you could get more energy out of them than sugar. How does the human body store spare energy? I guess it makes triglycerols and then stores them in adipocytes. Does the structure and function of triacylglycerols? Describe the structure and function of triacylglycerols. Well, there's a glycerol component bonded by ester bonds, esterification. Is that right? Ester bonds? Ester linkages to 3 fatty acids, usually of different types. What bonds are broken during saponification? The ester bonds are broken during saponification. Why does soap appear to dissolve in water, and how is this fact important to cleaning? It makes little micelles, so hydrophobic substances can hide on the inside, hydrophilic will remain on the outside, and overall it looks like everything dissolves pretty well. Let's take a look at those answers. 5.3, the human body, number 1, the human body stores energy as glycogen and triacylglycerols. Triacylglycerols are preferred because their carbons are more reduced, resulting in a larger amount of energy yield per unit weight. Triacylglycerols are preferred because their carbons are more reduced. So they're not oxidized, they're reduced. Let's write down that fats are more reduced than sugars. Glycogen is not a sugar, is it? What is glycogen? Multi-branch polysaccharide of glucose. Right. Number 2, triacylglycerols. Did I finish that one? In addition, due to their hydrophobic nature, triacylglycerols do not need to carry extra weight from hydration. All right, number 2, triacylglycerols, also called triglycerides, are composed of glycerol backbone esterified to 3 fatty acids. 3, the ester bonds of triacylglycerols are broken to form glycerol molecule and the salts of fatty acids. 4, soap appears to dissolve in water because amphipathic free fatty acid salts form micelles with hydrophobic fatty acid tails toward the center and carboxylate groups facing outward toward the water. Fat-soluble particles can then dissolve inside micelles in the soap, water, solution, and wash away. Water-soluble components can freely dissolve in the water. Great. Discrete practice questions. Number 1. Which of the following is not a type of glycolipid? Gangliosides, cerebroside, globoside, sphingomyelin. I guess this, is it the sphingomyelin? Glycosphingolipid, globoside, cerebroside, ganglioside are all the glycolipids. Sphingomyelin is not. It's a phosphate, phosphatidylcholine. It's a phospholipid, not a glycolipid. 1 is D. 2. During saponification, triacylglycerols undergo a condensation reaction. Triacylglycerols undergo ester hydrolysis. Fatty acid salts are produced using a strong acid. Fatty acid salts are bound to albumin. The ester hydrolysis sounds right because we're breaking up that ester bond. So let's go with B. 3. Which of the following best describes the structure of steroids? 3 cyclopentane rings, 1 cyclohexane ring. 3 cyclohexane rings, 1 cyclopentane ring. I think that's the one. 4 carbon rings differing in structure for each steroid? No. 3 cyclic carbon rings and a functional group. So we're going to go with B. 4. Soap bubbles form because fatty acid salts organize into lysosomes, micelles, phospholipid bilayers, hydrogen bonds. Soap bubbles I think they're getting at micelles, but soap bubbles are air-filled bubbles with water with a high surface tension making the outside of the bubble. That's probably the glycerol part. That's glycerol making water more viscous. I mean, so when you use soap, I imagine that the fatty acid part is in solution in the water that you're washing away so you don't see it. The bubbles that come out of that, like the water or the air bubbles that come out, that's air on the inside and just glycerol and water on the outside. But what they're getting at is micelles, right? Fatty acid salts organize into micelles. I think they're confusing this micelle structure for a soap bubble. This micelle structure is microscopic and you can't see it. A soap bubble is quite visible. I don't know what they're talking about. Number 5, steroid hormones are steroids that 1. Have specific high affinity receptors. Yeah. 2. Travel in the bloodstream from endocrine glands to distant sites. Yeah. 3. Affect gene transcription by binding directly to DNA. Steroid hormones probably don't, right? They probably cause a cascade of cyclic AMP or something. The peptide hormone is the one that can go into the DNA, I think. Let's double check that. Yeah, we're talking about fat-soluble stuff, so yeah, steroid hormone. Does steroid hormone go into the cell? No, peptide hormone goes into the cell. Amino acid-derived hormones and polypeptide are not lipid-derived and therefore cannot diffuse through the plasma membrane of cells. Okay, so peptide hormones stay on the outside of cells. Lipid hormones can enter the cell. Do lipid hormones interact with DNA? They can bind to DNA. They can get all the way into the interior of the cell. Great. No, interaction of steroid hormone receptors with DNA. Steroid hormones bind to their corresponding receptors in the cytoplasm of responsive cells, and this complex is translocated to the nucleus. So, not 3, but 1 and 2 sounds pretty good. C, 1 and 2 only. Six, why are triglycerols used in the human body for energy storage? They are highly hydrated and therefore can store lots of energy. B, they always have short fatty acid chains for easy access by metabolic enzymes. The carbon atoms of the fatty acid chains are highly reduced and therefore yield more energy upon oxidation. That sounds pretty good. Or D, polysaccharides, which would actually be a better energy storage form, would dissolve in the body. 6, C. We discussed that triacylglycerols are highly reduced, yielding a lot of energy when they are oxidized. Seven, which of the following is correct about fat-soluble vitamins? Vitamin E is important for calcium regulation. I think that's probably D. Vitamin D protects against cancer because it is a biological antioxidant. I think that is probably vitamin E. 3, vitamin K is necessary for the post-translational introduction of calcium binding sites. We did learn, they did say that it had something to do with post-translational insertion, right? Is it of calcium binding sites? Sounds plausible. Let's take another look at that. Vitamin K, calcium binding sites on calcium-dependent proteins. Great. That sounds good. Effect. Let's see. Vitamin A is metabolized to retinol, which is important for sight. That's also true. I think 3 and 4 both look pretty good. 7, C. 8, which of the following is true of amphipathic molecules? They form protective spheres in any solvent, with hydrophobic molecules interior and hydrophilic molecules exterior. They have 2 fatty acid chains and a polar head group. They are important to the formation of the phospholipid bilayer and soap bubbles. They have a glycerol base. So D is not always true. B is not always true. Those are 2 different cases. They do form these protective spheres, but hydrophobic being interior and hydrophilic being exterior, that's just specific to a polar solvent. It would be the opposite in a nonpolar solvent. So C is the most accurate. They are important to the formation of both the phospholipid bilayer and soap bubbles. Nine, which of the following is true about sphingolipids? They are all phospholipids. Some sphingolipids are not phospholipids. 2, they all contain a sphingosine backbone. What part of it was a sphingosine backbone again? Yeah, I guess that's true. This blue part is the sphingosine. They can have either phosphodiester or glycosidic linkages to their polar head groups. That is also true. 2 and 3 both look pretty good. Let's go with C. I want to look back and make sure in this chart that the sphingolipid backbone is the blue part. Sphingosine backbone. Yeah. All right. 10. Which of the following statements about saturation is false? It can describe the number of double or triple bonds in a fatty acid tail. It determines at least one of the properties of membranes. More saturated fatty acids make for a more fluid solution. I think that's the one that's false. Fully saturated fatty acids have only single bonds. That's true. It can describe the number of double or triple bonds in a fatty acid tail. I guess so. Yeah, triple as well. It determines at least one of the properties of membranes. Yeah, how permeable it is, right? So more saturated fatty acids make for a more fluid solution. I think unsaturated will make for a fluid solution. 11. Which of the following is true about glycerophospholipids? Glycerophospholipids can sometimes be sphingolipids. Yes. Depending on the bonds in their head groups. What is glycerophospholipid? That's Glycosphingolipid is the same thing as glycerophospholipid or no? Glycerophospholipids. So they're the sphingomyelins, are glycerophospholipids. Glycerophospholipids can sometimes be sphingolipids depending on the bonds in their head groups. Glycerophospholipids are merely a subset of phospholipids. I think that is true. C. Glycerophospholipids are used in the ABO blood typing system. ABO blood type, we learned, was I didn't write that down. I thought I wrote at least like signaling or something. Where was that about ABO blood type? Sphingolipids in general. It says they're sphingolipids, but then it says Like glycerophospholipids, sphingolipids are also sites of biological recognition. Glycerophospholipids have one glycerol, one polar head group, and one fatty acid tail. Glycerol, polar head, and one fatty acid tail. Yeah, that looks right to me. So sphingolipids have a sphingosine backbone. Glycerophospholipids have a glycerol backbone. So glycerophospholipids are not sphingolipids. To me, they seem fundamentally different, huh? So, A is false. Glycerophospholipids are phospholipids. That's true. Glycerophospholipids are used in ABO blood type. No, sphingolipids are used in ABO blood type. Glycerophospholipids have one glycerol, one polar head group, and one fatty acid tail. That looks right. Oh, one fatty acid tail. Two fatty acid tails, not just one. So D is also false. So we'll go with 11B. It's a subset of phospholipids. It is a phosphodiester bond there. So it is a phospholipid. 12. Which of the following statements about terpenes is false? Terpenes are strongly scented molecules that sometimes serve protective functions. B. Terpenes are steroid precursors. C. A terpene is made of 3 isoprene moieties and therefore have 15 carbons. D. Terpenes are made of plants and insects. Made by plants and insects. D sounds fine, true. C, a terpene is made of 3 isoprene units. Let's double check those isoprene units. Triterpene is, I think, 6 isoprene units. 30 carbons. So C is false. Are terpenes steroid precursors? Yeah. Cholesterol and steroids. So true, true, false, true. 12 is C. 13. Which of the following is true about cholesterol? Cholesterol always increases membrane fluidity in cells. Cholesterol is a steroid hormone precursor. Cholesterol is a precursor for vitamin A, which is produced in the skin. Cholesterol interacts only with the hydrophobic tails of phospholipids. So that vitamin A is produced in the skin, that's probably vitamin D, so maybe that's what makes it false. D, cholesterol interacts only with the hydrophobic tails of phospholipids. Cholesterol has a hydrophilic region too. This alcohol over here. So D is false, C is false. It is a steroid hormone precursor, but it doesn't always increase membrane fluidity. It can decrease membrane fluidity in high temperatures. So 13 is going to be B. It's a steroid hormone precursor. Great. 14. Which of the following statements regarding prostaglandins is false? Prostaglandins regulate the synthesis of cyclic AMP. I think that's true. Prostaglandin synthesis is inhibited by NSAIDs. I think that's also true. Prostaglandins affect pain, inflammation, and smooth muscle function. Also true. Prostaglandins are endocrine hormones like steroid hormones. I think that's true too. They all sound true to me. What are prostaglandins again? Where are they? Prostaglandins. Do they regulate the synthesis of cyclic AMP? Is to regulate the synthesis, right? So B is true, or A is true, B is true, C is true. Prostaglandins are endocrine hormones. I guess they're produced by all cells. Are produced by almost all cells in the body. So we don't have to call them endocrine hormones. So let's go with D. Prostaglandin. Yeah, they're not hormones. They're lipids. Having hormone-like effects, but not necessarily a hormone. 15. Which of the statements regarding waxes is false? Waxes generally have melting points above room temperature. Sounds good. B. Waxes are produced only in plants and insects and therefore must be consumed by humans. I think that's false. We produce wax. C. Waxes protect against dehydration in parasites. True. D. Waxes are esters of long-chain fatty acids and long-chain alcohols. Sounds good. B is the only one that is false. Let's check our answers. 1 is D, 2 is B, 3 is B, 4 is B, 5 is C, 6 is C, 7 is C, 8 is C, 9 is B, not C. Sphingolipids can either have a phosphodiester bond and therefore be phospholipids, or have glycosidic linkage and therefore be a glycolipid. Not all sphingolipids have a sphingosine backbone, as in statement 2. Some have related sphingoid compounds as backbones instead. So they went with B, which was 3 only. They can have either phosphodiester or glycosidic linkages to their polar head groups, but that they all contain a sphingosine backbone is not true. Some have related sphingoid compounds as backbones instead. And we learned that sphingoid meant oxygenated or modified sphingoid. Let's double check. Maybe I forgot to write this, but sphingoid I guess they don't get into the sphingoid. All of these examples have sphingosine backbones. All right. So 9 is B, 10 is C, 11 is B, not D. Glycerophospholipids are a subset of phospholipids, as are sphingomyelin. Sphingomyelins are also phospholipids. Glycerophospholipids are never sphingolipids because they contain a glycerol backbone rather than sphingosine, or a sphingoid backbone, eliminating choice A. Sphingolipids are used in the ABO blood typing system, eliminating choice C. Glycerophospholipids have a polar head group glycerol and 2 fatty acid tails, not as in choice B or D, so the answer is B. Oh yeah, not D. It's not 1 fatty acid tail, it was B. Why did I say D? It's a subset of phospholipids. Great. 12 is C, 13 is B, 14 is D, 15 is B. And that is it for lipid structure and function. Next time, Chapter 6, DNA and Biotechnology. Thanks for watching. I'll see you next time.